Now we're going to take a look at how the information gets from the retina into the brain and how we process that visual information. John Medina, in his book Brain Rules, if you've ever read that, he describes a study in which wine experts were asked to describe the wine they were tasting. Now, apparently, there are different words used to describe red wines versus white wines. And I guess if you're an expert, you're never supposed to cross those two vocabularies. So, in this study, researchers wanted to see what would happen if they colored white wine red with odorless, tasteless dye. Would the wine experts know the difference? Well, out of 54 subjects, all of them used the red wine vocabulary when they were actually drinking white wine that was dyed red. So, what does this mean? Well, it means the visual inputs overrode their highly trained palates and noses. And really, the point that Medina is making is that vision trumps all other senses. That's why there's so many chapters in your text devoted to vision and why we spend so much time on it. Think about it. The information that your brain can receive from what you're looking at is far more complex than the other senses. And so that visual processing, those structures in the brain, there's quite a bit of real estate devoted to that. Really, researchers have identified over 30 areas of the brain that are involved in visual processing. But don't worry, we're not going to talk about all 30. We're just going to talk about the main areas. So it's not surprising that we really see with our brains and not our eyes. If you remember from chapter 1, people with damage to the primary visual cortex, which we call area V1, can suffer from cortical blindness. And with cortical blindness, the individual has normal eye function. So it's possible to have nothing wrong with your eyes, but because there's something wrong in the brain, you have blindness. With cortical blindness, the individual has a normal pupil response, normal eye movement, normal retina, but they lose the ability to perceive light and dark. They also lose something called the menace reflex. So with typical vision, if somebody throws a ball right at your eye, you're going to react. You're going to blink. The eye automatically detects a menace and tries to protect itself. Well, there's a rare form of cortical blindness that's called visual anosognosia, or Anton Babinski syndrome. It's a rare neurological condition in which patients deny their blindness and affirm adamantly that they can see. The clinical presentation includes memory distortions and sometimes confusional states. So these patients, if they're pressed, they'll actually make up what we call confabulate memories, but they are not being they're not being untruthful on purpose. That's just the way that they accommodate the situation. So they will even, pretend to read a newspaper and tell you that they can read the newspaper. So it distorts their memory processes. We're not quite sure why this happens. One hypothesis is that when that damage to the visual cortex in this particular syndrome, it results in the inability to communicate with the speech language areas of the brain. So, in other words, the person can receive the visual image, but can't interpret the visual image. So the speech centers of the brain confabulate a response. There's also something called visual neglect. And this is where a patient can actually see the entire visual field, so can see with both eyes, but somehow ignores the objects on one side. So if you ask them to draw a diagram of what they see, they'll only draw one side of what's in their visual field. Or they might, for example, a man with visual neglect might shave only one side of his face. So we think this is due to a lesion of the brain, like a stroke, most often in the right cortex. And the patient, although conscious of objects in the left visual field, does not pay attention to them. So the impairment occurs in the posterior parietal lobe, which receives projections from the primary visual cortex. So these are just a few examples of the problems we see with vision because of problems with the brain, not with our eyes. And over the next few weeks, we'll look at other disorders. For now, we want to get to a basic understanding of how the brain processes visual information. We know that the visual pathways begin in the retina. Photoreceptors transduce light in the retinal image into neural signals that propagate through neural circuits within the retina. Lateral inhibition in neural circuits underlying the RGC's receptive fields promotes representation of edges at locations in the retinal image. It also signals when there are abrupt changes in brightness. So it's the RGCs that take the information transduced by the photoreceptors to the brain via the optic nerve. Signals generated by the RGCs carry information about edges and other visual features. They bundle together and then they form the optic nerve, exiting each eye and carrying signals to the brain. This information travels to the primary visual cortex, or V1, and then information from the V1 travels along two different pathways called the where pathway and the what pathway. And let's see an example of this. Sorry, my computer is being slow. So this is just a brief clip of the where and the what pathways.
There's just a split second to see the bee. Even though it takes place right in front of her eyes, she can't see it directly. The retina must first transform the light beams into nerve impulses. The brain then computes those impulses and reconstructs the image she's able to see. First, the impulses reach the primary cortex. Nerve cells are very responsive to curves and contours, where light transitions to dark. Now, two processes take place at once. The process above controls what you see, below, where you see it. The where process might define the location of an object. Its shape is irrelevant. What's significant is that the image must be three-dimensional. Certain neurons respond to motion. Speed is calculated exactly, but the movement's actual location is only approximate. While one part of the brain only deals with the where, the other part analyzes the what. The brain detects an outline and quickly determines an object's shape. The result is then compared in the brain's memory. If the object has been seen before, it's easier to recognize. Most people or objects are identified this way. It takes longer to analyze and reconstruct unknown objects. Meanwhile, other parts of the brain determine color. In a split second, the brain extracts the relevant information from the blast of neurons. But how it combines the different features is unknown, and it happens all at the same time. From there, the information is reported to the primary cortex. It's the cortex that actually perceives the image, and after a quarter of a second, she can react accordingly.
Okay, I'm not sure I would eat that bagel after a bee had been on it, but maybe that's just me. I don't know. Um, okay, so we have these two different pathways. So let's break down this process and first look at how do we get the signals from the eye to the brain. So neural signals originating in the brain travel along pathways deeper into the brain through increasingly complex networks. And they have characteristics or organizational principles that define these pathways and networks. The first is functional specialization. So there's specialization of different neural pathways in different areas of the brain for representing different kinds of information. And that just makes it more efficient. And then really important is this concept of retinotopic mapping. So we see this retinotopic mapping in the way the brain is structured, the visual cortex is structured. Arrangement of neurons in the visual system whereby signals from retinal ganglion cells, those RGCs, with receptive fields that are next to each other on the retina, well, they travel to neurons that are next to each other in each visual area of the brain. So there's a correspondence. That's why we call it a mapping. So the visual area in the retina is mapped onto a similar area in the visual areas of the brain. And we see this in terms of the functional areas, but also when we look at the cell structures. So there are main pathways from the retina to the brain, and first would be the optic nerves. We talked about that, how the RGCs, the axons, bundle into the optic nerve. They leave the eye, so you have through the optic disc, that blind spot area, they leave the eye and they travel to the optic chiasm. So get my mouse here. So here we have the optic disc and we have information that's traveling along to the optic chiasm. Now, the optic chiasm is the location where the optic nerves from the two eyes split. So half of the axons from each eye go left, and the other half of the axons from each eye go right. So again, if we look at this, they're determined by the red and blue lines. So here we have information from the left nasal retina, because it's closest to the nose. So that information goes through, gets split, goes in this direction. The left temporal area, that information gets split, goes in this direction, and the same for the other eye. So in the optic tract, we have the continuation of the optic nerve past the optic chiasm. So the right optic tract will have the axons from the right RGCs that are in the right half of the retina of each eye. So the right half of the retina of each eye is going to be the red. And then the opposite is true. The left optic tract has the axons from the RGCs and the left half of the retina of each eye. So that's what you're seeing in the blue. Neural signals carrying information from the left half of the visual field, depicted in red, are sent via the lateral geniculate nucleus and the optic radiations to the primary visual cortex in the right hemisphere. So we can see this traveling along. Here's the LGN, the lateral geniculate nucleus. We just say LGN. And the optic radiations from there travel to the primary visual cortex. Sometimes from the optic chiasm, some of the information will go to the superior colliculus. Okay, so we see that when it's coming from the left side, so the left nasal retina, the red track actually goes to the right side of the brain. So when you have your left visual field, what you're looking at in the left visual field is being processed by the right side of the brain. What you're looking at in your right visual field is being processed by the left side of the brain. And we call that contralateral organization, which just means opposite side. So we see that a lot. If you have brain damage, for example, on the right side of your brain that interferes with mobility, that interference will be experienced on the left side of your body. So signals carrying information from the right half of the visual field depicted in blue, which would be those signals from the left temporal retina and the right nasal retina, are sent to the left hemisphere. And as I said, in each hemisphere, some pathways branch off from the optic tract and go to the superior colliculus. And we'll talk about that in a minute. Superior colliculus. So the LGN is part of the nucleus, the lateral geniculate nucleus. It's part of it's part of the thalamus. So there are three structurally and functionally different layers. The first is called the magnocellular, or sometimes we just say M layer. And that's associated with dynamic visual properties like motion and flicker. And so here in this example, we have 1 and 2 are magnocellular layers. Now magno related to magna, which means large. So they have larger cells. The parvocellular or P layers are associated with static visual properties like color, pattern, texture, shape, depth. And so 3, 4, 5, and 6, those are the parvocellular layers. And then in between, and they're not numbered, but in between the different layers, 1 through 6, we have the coniocellular or K layers. We're not quite as sure about what those layers do, but it's thought that they probably play a role in color perception. So the cell bodies that are in the 2 magnocellular layers, layers 1 and 2, are relatively large, while those in the parvocellular layers, which are layers 3 through 6, are relatively small. And then the coniocellular layers are the lighter colored layers, and those cell bodies are too small to be seen in this image. Altogether, there are 12 layers in the lateral geniculate nucleus, or LGN, and we say the order is a retinotopic map because the location of cells in the LGN has an approximately map-like correspondence with the retina. So we're going to be seeing that over and over. So in other words, cells near one another in the LGN receive information from the photoreceptors near each other in the retina. The LGN is like the first gatekeeper in the brain for visual information. Researchers think that the LGN modifies visual information by using top-down feedback from higher brain structures, not just the primary visual cortex. So, for example, we know that attention influences the LGN and how it's operating. About 90% of the axons go to the LGN from the ganglia cells. Most of the remaining go to the superior colliculus. And the superior colliculus is similar to the LGN. So there are these multiple layers, and those layers are retinotopic. It sits on top of the brainstem, so there's one on each side. The superior colliculus controls eye movements and allows the brain to quickly know where things are. It's not as concerned with the what of things. It's not as concerned with color, shape, etc. So it just makes rapid use of the information coming from the retina. It doesn't need to go through the primary visual cortex. It can go straight from the retina to the superior colliculus. It also has connections between its layers, which allows it to integrate visual experiences with auditory experiences and touch. So it all has to come together, right? So we are talking to somebody, we're looking at them, we need to be able to hear them, or we need to be able to touch something that we're looking at and pick it up. So we call this multisensory integration. So the superior colliculus plays a role in multisensory integration. In the next video, then, we'll look at the primary visual cortex.